Hello, everyone. Welcome back from your sessions. So today we've been enjoying talks on visual signaling, visual systems, and on understanding the behaviors associated with optical signaling. And I'm very pleased to introduce our keynote speaker, Dr. Eleanor Cave, who will be bringing all these topics together to give us a big picture understanding on the mutualistic relationship between cleaner shrimp and their reef fish clients. Dr. Cave is speaking to us from her new lab at the University of California at Santa Barbara, where she is an assistant professor in the Ecology, Evolution, and Marine Biology department. Before starting as a professor at UC Santa Barbara, Dr. Cave did her PhD at Duke University with Tonka-Johnson. She later moved to a postdoctoral position as a Marie Curie Fellow at the University of Exeter. Her list of awards and achievements is long and includes being a National Geographic Young Explorer, as well as more recently winning the Jasper Loftus Hills Young Investigator Award. She's worked on a variety of organisms, including birds, fishes, and, as she's about to tell us today, shrimp. So please join me in welcoming Dr. Cave as she tells us, or she shed light on the ways cleaner shrimp don't get eaten by the predatory clients that they clean.
Wow, thank you so much. Okay, let me share my screen. First, you'll get a lovely view of the Tetons. Those are not cleaner shrimp. Okay, can everyone see that cleaner shrimp there? Great. Okay, well, first of all, thank you so much to the organizers of Living Light for the invitation to come and speak. This is such an honor. It's been really fun for me. This is the best early morning I've had in a very long time, hearing about all the amazing research done by all the early career scientists in this field. So today I'm going to tell you about work that I started during my dissertation. I put it away during my postdoc, and now I'm back to it as I start my own lab, which is what I'm doing right now. And indeed, we don't have a ton of time, so I'm just going to dive right in. Okay, so what is a cleaner shrimp? Well, here's one. This is the Caribbean species Ancylomenes pedersoni. This is an image from Curacao. And cleaner shrimp are like the dental hygienists of a coral reef. So they live at a set location, which we call a cleaning station. And then fish, which we call clients, come to the station, pose in the substrate nearby, and then allow the shrimp to hop on and pick off ectoparasites and dead skin. And one thing that's really intriguing about this interaction is that at the site in Curacao, as well as others around the globe, we see that half or more of visits from client fish are by species that can and regularly do eat crustaceans. So this raises the question, why doesn't the client eat the cleaner? Well, one hypothesis is that visual signals have evolved, which identify cleaners as beneficial partners rather than a food item, and identify clients as seeking cleaning rather than a meal. And as a visual ecologist, this is an interesting hypothesis to me. So if we want to start exploring the question, how do cleaners and clients perceive one another? We need information on cleaner shrimp vision, client fish vision, and those potential signaling behaviors in nature. So for my talk today, we'll start by talking a little bit about what we know about cleaner shrimp visual capabilities. I will touch so briefly on the enormous literature that there is on client fish vision. And then we'll apply those measures of visual capability to interactions in nature to talk about what behaviors might or might not serve as visual signals. And then I hope at the very end to have just a minute to tell you about some brand new work that I think will be especially interesting to this crowd about the ultrastructure of these, the body parts that cleaner shrimp are actually using to signal. Okay, so starting with those visual capabilities, one of the first things I did back when I started my PhD was team up with Tammy Frank at Nova Southeast University to look at spectral sensitivity in cleaner shrimp. And I think it will become clear why we did that when I show you the pictures of the species we used, which are Lysmata amboinensis, Ancylomenes pedersoni, and Urocaradella antembrunii. And as you can see, these are all beautiful, colorful, finely patterned animals. We picked these 3 species because they are from different genera that each have cleaner shrimp. They're actually all from different geographic regions as well. And we thought this was going to maximize our chances of finding what we were pretty much sure was going to be interesting variation in their color vision capability. So using electroretinography, actually, we found evidence for only a single spectral sensitivity peak in all 3 species. For those of you who are familiar with ERG data, this is dark adapted data. We also did 3 additional chromatic adaptations that all supported this finding. This little tail over here in the UV is very likely just the beta peak of the visual pigment. So this was evidence in which allowed us to conclude that these are actually monochromats, so that they don't perceive all that color that they themselves display, which was kind of surprising to us given that they are colorful, they live in colorful environments, they interact with reef fish, which are famously colorful. But okay, color is not the only aspect of a visual scene. And in fact, my favorite visual parameter is visual acuity. I've seen a lot of mentions of visual acuity at this conference today, which has made me so excited because it's really such an interesting parameter. It's highly, highly variable across species. So this plot is showing us just a handful of the species for which we have data. We have eye diameter on the x-axis, acuity on the y, and acuity is measured in or displayed in units called cycles per degree. Don't worry too much if you don't know exactly what those units are. Just know that higher numbers means finer spatial vision, ability to see smaller details. So I just want to linger on this graph for a moment and point out a few things. So first of all, we see that acuity is presented on a log scale. So it's varying by at least 4 orders of magnitude across animals that we consider to have image-forming eyes. There's also a strong and positive relationship between acuity and eye size. And because eye size itself is tightly correlated with body size, that means we tend to see the highest acuity in large animals with large eyes. Compound eyes, as many people have pointed out today, are acuity limited because of their optics relative to camera eyes. Those animals in red are those with compound eyes, and indeed they're falling out at the lower end of the acuity spectrum. And then just out of interest, I'll point out that here's us up at nearly the very top, which should be like really exciting for everybody here because humans aren't really the pinnacle of any sensory modality at all. But here we are with some of the finest spatial vision in the animal kingdom. And it also should lead all of us who study any kind of visually guided behavior to think about what that means in terms of our own study species and how they're perceiving spatial detail. So in terms of measuring acuity, there are a variety of methods. We used a behavioral method called an optimotor, as well as a morphological method, just looking at the diameter of the ommatidia to estimate acuity in the same 3 species of cleaner shrimp. And here they are at about 0.1 cycles per degree, which is pretty low, as you can see, compared to other animals. So, okay, to recap what we've learned so far. We've started with our beautiful, colorful, finely patterned cleaner shrimp and found that very likely they are in fact monochromats, and their acuity is really shockingly low. This image on the right is made with an R package that Sanka Johnson and I wrote called Acuity View. And Acuity View just uses Fourier methods to delete spatial frequencies from an image that should be unresolvable to a viewer of a given acuity from a given distance. And the main caveat I really wanted to emphasize here is that this is very unlikely to show us what the animal actually sees for a couple of reasons, right? These are static, so motion is always going to make things more apparent. And they also, we can't account in something like Acuity View for post-processing, edge sharpening and contrast enhancement and so on. So this isn't necessarily a representation of a cleaner shrimp's perceptual world, but what Acuity View can tell us is what spatial information likely makes it past the photoreceptor array for post-processing to even work on. So these were unexpected findings for me as a first-year PhD student. I had looked at these animals with my own human visual system and biases, and I thought these flashy color patterns really must serve some kind of interest-specific signaling purpose, like species ID or in agonistic encounters. But from these results, I think it's fair to say that's probably unlikely. But unexpected findings can lead us to unexpected lines of inquiry. And in my case, this actually opened up two lines of thinking that have really guided my research ever since. So the first is that indeed, learning about this animal's spectral sensitivity did give us some insight into its perceptual world, but learning about its acuity gave us additional very important information. And this is even an incomplete picture in that, like, we haven't built in things like temporal resolution, et cetera. But it really drove home for me that it's important to consider multiple aspects of visual capability if we want to think about perception. Second, the fact that it's so unlikely that these color patterns serve an intraspecific signaling function made me think about who else these cleaner shrimp are interacting with, and that is, of course, the client fish. So client fish are the other half of our story. So let's just really briefly talk about client fish vision. Fish and reef fish in particular have famously diverse visual systems. A lot of people have done really excellent work on that. The work that I've been involved in was really led by Dr. Lori Schweikert, who's now at University of North Carolina, Wilmington. And she synthesized published literature on 213 species of ray fin fish, their color vision capabilities as measured by MSP. And from that database, we could see that all the reef fish, those fish that are likely to serve as clients, tend to be tri, tetra, or pentachromatic. But again, this is kind of an oversimplification. But for the purposes of today's talk, we'll just assume that any fish that can serve as a client likely perceives color, discriminates color as well as we do, if not better. And I know that's an oversimplification, but it's a short talk. For acuity, we took a similar approach. So I synthesized the published literature I could find on acuity in fish, found data on 159 species. Acuity in fish overall ranges from about 1 to 35 cycles per degree. Even that high value, 35 cycles, that's in large predatory fish like sailfish, rock bass. That's still only about half as fine as human vision. And then if we focus in on those reef fish that are likely to serve as clients, we see that their acuity is between about 1 and 14 cycles per degree. So we've seen so far then that cleaner shrimp have monochromatic low resolution vision, whereas client fish have good, for lack of a better word, color vision and acuity that's at least an order of magnitude higher than a cleaner shrimp. So this is an interaction that's occurring between two parties with very different visual capabilities. So let's take that information about visual capability and apply it to some observations of interactions in nature and think about what's actually being perceived as these parties are interacting with each other. Okay, so just as a reminder of what happens in a typical cleaner client interaction, this is in Ancylomenes pedersoni in the Caribbean. The first thing that usually happens when a client fish is approaching a cleaning station is the cleaner shrimp will whip its antennae, which I'm showing you here on this kind of looped GIF. And so that is usually the first step that initiates the cleaning interaction. Then we see the client adopt this cleaning solicitation pose. They'll go totally motionless in the substrate. By the cleaning station, they'll often flare out their opercula, open their mouths, things like that. And then often we see them change from the color morph that they arrived in to a darker color morph. And this color change is rapid. It happens in 5 to 10 seconds. At which point cleaning occurs. Sometimes during cleaning, client fish will jolt. It looks almost like they got pinched. And cleaning can last anywhere from a few seconds to more than a minute. But eventually, the client will return to its lighter color morph, and usually pretty shortly after that, it exits the scene. So the two behaviors that we're interested in looking at for signal function in particular are that antennae whipping motion by the cleaner shrimp and that color change to a darker morph by the client fish. And that's in part because there's a lot of literature on this species that had referred to both of these behaviors as signals in the past, but without actually explicitly testing them for signal function as such. So what do I mean by signal function, right? Well, we need a working definition of a signal if we want to decide whether something is or is not a signal. And for any animal behaviorists in the audience, you know that this is actually a pretty hotly debated topic, and classifying things as signals or not is an active area of research. But I think there are a few criteria we can hopefully all agree on that something has to fulfill in order to be considered a signal. So for an act or structure to be a signal, it must predict a state change in the receiver. By that, I mean, it has to be followed by a reliable receiver response. Basically, because if no one's ever responding to your signal, the signaler should stop sending it and the system should break down. Signals must also convey information about the sender's state or future behavior, by which I mean the receiver has to be able to extract some useful information from the signal, because otherwise they'll stop responding to the signal and the signaling system should break down. We also know that the outcome of a signaling interaction must, on average, benefit both the sender and the receiver, because this is an oversimplification, but basically if the outcome is consistently negative to one party or another, they should stop participating and the system should break down. Okay, so these are criteria that come from a lot of theoretical and empirical work on signal theory. And to this, I would add just one more, which is not a novel thought by me, but that's that signals must also be perceptible by the receiver. That's something that's probably obvious to this audience, but as an animal behaviorist, this isn't something we explicitly test as often as we maybe should, given what we know about the diversity of sensory capabilities in the animal kingdom. We bring our own human biases, our own human perception to our questions and our hypotheses sometimes, and it's worth asking, is that thing that I think is flashy and exciting actually perceptible by the receiver? And if so, how are they perceiving it? Okay, so we can use these criterion to make some predictions about our potential signaling traits of interest. So for example, if that antennae whipping motion by the cleaner shrimp is a signal, it should predict a state change in the receiver. And specifically, we predict that client fish that view antennae whipping adopt that cleaning solicitation pose, because that's physically necessary for cleaning to begin. We also predict that antennae whipping should convey information about the sender's state, or in this case, its future behavior, specifically that cleaner shrimp that antennae whip do then go on to clean. Also, the outcome of any signaling interaction should be something that benefits both the sender and the receiver. And antennae whipping should be perceptible by a variety of reef fish visual systems, all of which can serve as clients. For today's talk, we'll just assume that anything that ended in cleaning did benefit both the sender and the receiver. As with everything in biology, it's never quite that simple. And we know that cleaning can actually range from a little bit costly to neutral to mutualistic, but in general, the outcome of a cleaning interaction is beneficial to both parties. Okay, so to test these predictions, we went to Curacao. We deployed GoPro cameras at cleaning stations, and we recorded and then annotated 133 hours of footage from 8 cleaning stations. And in that footage, we had 199 interactions between 10 species of clients and 18 individual shrimp. So after we had done all these behavioral annotations, what we had for each of these interactions is basically a sequence of behaviors that occurs as cleaners interact with clients. And one way that you can analyze such sequences is using a network analysis approach. So some of you are probably familiar with networks, but in case anyone isn't, I'll just walk you through how they work. So in a behavioral network, the first thing you have are your network vertices, these circles. So these are the behaviors in your interactions. So we have our cleaner behaviors in yellow, client behaviors in blue, and the size of the circle is scaled to represent the number of times that behavior was observed in a given data set. And then in between your network vertices, you have what are called transitional probabilities. So this is the probability that once a behavioral sequence is in a given state, the next behavior in the sequence is this or that. So for example, all of our sequences begin when the client enters the camera's field of view, at which point there's a 70% probability that the next behavior in the sequence is antennae whipping by the shrimp, 29% probability that the next behavior in the sequence is posing by the client. You'll notice those don't add to 100, and that's because I'm only going to show you statistically significant transitions today. Now, real networks have a whole lot more than 2 transitional probabilities. Don't worry too much about all the detail in this figure. For me, building this figure was fun and a little bit surprising because I thought of this as a relatively simple interaction with not many behaviors on the part of each party. But it was interesting to see how many different pathways there are by which these interactions can proceed. Okay, so we can use networks like this one. So this was built using the 144 interactions in our data set where antennae whipping occurs. And we can use these networks to look at our predictions about signal function. So from this network, we can see indeed antennae whipping here does predict receiver behavior in that it's followed by posing 80% of the time. And in fact, the only significant transition away from cleaner shrimp antennae whipping is to posing by the client. We can also see from our behavioral sequences that antennae whipping predicts the cleaner's behavior. So actually 80% of cleaners that antennae whip do then go on to clean through some pathway or another. And as far as these signals being perceptible, in terms of acuity at least, we were able to make little acuity view movies, slow down to half speed, using the highest and lowest known acuity in any reef fish. This is assuming an interaction distance of 30 centimeters, which is a little bit further than most of the interactions in our videos. And you can see that at least in terms of acuity, especially once we've built motion into the picture, it seems as though antennae whipping should be resolvable, even to reef fish with low acuity. Okay, so antennae whipping does seem to predict that clients will adopt that cleaning solicitation pose. It does predict whether cleaners will go on to clean. And again, in terms of acuity, it does seem to be perceptible by a variety of reef fish. So it's probably a signal by these criteria. What about that color change behavior by clients? Well, we use the same network analysis approach that I've just described to look at color change, but instead of walking you through all those networks again, I'll just summarize for you that we found there are 2 pathways by which cleaning can occur in this species. So the one I just told you about is that usually when a client arrives, the cleaner shrimp whips its antennae, the client poses, and then there's cleaning. And we do see fish change color in this pathway at variable points in time. But about 20% of the time that clients arrive at cleaning stations, there's no antennae whipping. So no indication by the shrimp that it intends to clean. At which point clients can leave, but they actually do that very rarely. They can also pose anyway and not color change, or they can pose and change to a darker color morph. So for those clients that pose and don't change color, they have only a 15% chance at that point that they'll get cleaned. But those clients that pose and change to a darker color morph, more than triple the chances that they'll get cleaned to 50%. So this could indicate that color change is a signal that clients can use themselves to elicit cleaning, especially in cases where the cleaner hasn't already indicated its intent to clean. Which I find really exciting because it's somewhat of an indication of a sort of stable communication system between cleaners and clients. But you might be saying, that is really exciting, but you spent the beginning of your talk telling us that cleaner shrimp have monochromatic low resolution vision. So what good is a color change signal? And that's where we can think about the fact that, I've been showing you goatfish because they are lovely clients. They turn from white to this like dark red color. But across client fish species, we see a really consistent pattern where if they're going to color change, they do so by going from a lighter morph to a darker morph. So things like surgeonfish and tang will show up light blue and turn almost black. Snapper will show up white and adopt really thick, dark bars. So it is something that appears to be a color change to us, but it's always a change from a light to a dark. It occurs at very close range, and it doesn't involve any fine scale spatial stimuli. So this is something that ought to be perceptible by a cleaner shrimp. Okay, so it looks like in this species, antennae whipping is a signal, perhaps of intent to clean. Color change by clients likely signals desire to be cleaned, but there's just so much work to be done on this color change behavior in client fish. But overall, if neither party signals, if there's no antennae whipping and no color change, cleaning only occurs in 2% of interactions. So these might not be completely visually mediated interactions, but these visual signals seem to play a pretty important role in whether or not cleaning actually occurs. Okay, so just at the very end here, I'm going to tell you about something very new, looking at the ultrastructure of those cleaner shrimp signaling structures, the antennae in this case. And to introduce this, I have to tell you very quickly that after doing this project on Ancylomenes pedersoni in the Caribbean, I went to the Red Sea, where I was looking at Lysmata amboinensis. This is work I did in collaboration with Catherine Chen, who was an undergrad at Duke at the time. And you can see in this video, Lysmata amboinensis has these white front legs that it's rocking back and forth in the presence of clients. It's also got long white antennae that it's waving around and tapping on the client with. And Catherine used those same network analysis techniques to show that indeed, these are signals, but actually Lysmata amboinensis is only using them in the presence of potential predators. So it actually exhibits this whole suite of behavioral modifications where it's essentially signaling to potential predators and cleaning them less. So it's modifying its behavior based on the risks of interacting with specific clients. And this was really interesting to me as an animal behaviorist to think about these two shrimp in different families, in different geographic regions, independent evolutions of the same cleaning behavior. There were some behavioral differences indeed between the species, but one striking similarity is that they're both using these white body parts, waving them around in the presence of clients. And in fact, anecdotally, this is what we see in a variety of cleaner shrimp species from around the globe. So signal function hasn't been demonstrated in all of these, but we see that cleaner shrimp tend to have these white body parts and they wave them around in the presence of client fish. And this struck my collaborators, Alex Davis and Sanka Johnson at Duke, in part because these signaling traits actually remain so white, even at depths of 10 or 20 meters, they're really popping and looking quite bright. And so they were curious what might underlie that real bright signal that we're getting off these antennae. And this is very much Alex's work. So his work, his results, and I'm very lucky he's letting me give a sneak preview here. But he did some scanning electron microscopy and found in each species antennae this layer of spherical nanoparticles about 1 to 3 micrometers thick, each particle about 300 to 400 nanometers in diameter, just inside the cuticle. And he thinks these may be isoxanthopteran. This is based on particle size, a likely refractive index based on the amount of light that they scatter. And as we've heard in other talks just today, isoxanthopteran is present in the tapeta of many decapod crustaceans. So this is a reflective compound already present in their eyes that may also be in these highly reflective signal structures. The size of these spheres is at an inflection point for reflectance. So they're not actually the optimal size for reflecting the most light, but they are at this inflection point where increasing the size has very little effect on reflectance, but decreasing it has a dramatic effect. So overall, Alex's modeling work is suggesting this layer might be effective in enhancing the brightness of the antennae. And whether this is some kind of signal boosting structure, we have a lot of work to do. This is just the start. But particularly looking at some comparative work about whether this is found just in cleaners or also related non-cleaners is something that's particularly interesting to me. So this is just a whirlwind tour of what's going on with cleaner shrimp. This has set up some work I'm really excited about doing next, digging into the evolution of these signaling traits, the sensory ecology of these signals. You know, if this is convergence on white signals in cleaners, why is that such an effective signal to client fish? And there's just so many unanswered questions, and it's a really fun system to be working in. So with that, I have people and funding to thank, and just a quick plug for the fact that my lab is brand new, and in the next year, I will be recruiting a postdoc. So if the work I've described here today is interesting to you, please reach out and I'll keep you updated about that search. And I would be happy to take any questions. Thanks.
Thank you very much, Eleanor, that wonderful talk. There are some questions in the chat.
Okay.
I can read them to you. Okay. Okay, from Nico, did you see such a color change more in species that tend to come to shrimp cleaner stations as a group rather than as a single individual? From my own observations, fish species that dramatically change to a redder color at cleaning stations with cleaner fish tend to be groups of mullets or fusiliers who take turns patiently. Only the individual who is currently being cleaned turns reddish and loses its reddish color quickly when leaving the station. This suggests that the signaling within the group may also play a role.
Yes, that is such a fantastic question. I think that's right. I mean, so my data set on the cleaner shrimp, or sorry, the client fish color change is limited to this data set from Curacao. And that is not maybe the greatest place to ask questions about the client fish behaviors because it's pretty overfished. And so it's a pretty depauperate client fish community. But you're right, it tends to be things like goatfish and they're showing up in groups. And as you say, patiently waiting turns. And honestly, that's such a great point that it might play a signaling role within the group. That's something that with the camera setup I've currently got, I wouldn't really be able to test because to see the antennae whipping and the cleaning, we have to zoom in so closely. But in the future, we're hoping to deploy multiple cameras so we can get kind of a bigger picture view of what's going on when groups of clients approach. And I think that's such an interesting hypothesis. That's, yeah, I'll definitely give that some thought. I really like that idea. Thanks.
Actually, how about Isabel, if you're there, would you like to actually ask your question or do you want me to read it?
Okay, yeah, it's fine. So can you hear me?
Yeah.
Hi, really nice talk. Sorry,
Thank
I'm
you.
from a different field, so for me everything is quite new. So I was wondering, so you mentioned that the shrimps are monochromes, etc. But have you checked the obscene complement?
No, it's a great question. So when we wrote that paper, this was the primary question by the reviewers, was like pointing out that it is obviously much harder to prove the absence of something than the presence of something. So they wanted us to look at additional methods for really proving that these are monochromats rather than maybe dichromats, which is common in crustaceans. And we just didn't have, the resources or the techniques really, I guess, in the know-how in our group to do that at the time. But it's such a good question, and something I'd be really interested in looking at is incorporating a couple of other methods to really confirm the color vision capability in these species and looking more broadly, right? There are dozens of species of cleaner shrimp around the globe, and I've only looked at 3. But I saw today a lot of people are studying crustacean vision. So maybe someone out there like really wants to help me with this particular question, which would be awesome. Yeah.
Well, I have another question, but
Sure.
because you mentioned that they use the antenna as a or to make a response. But do you think that as the antenna are the main chemosensory organs, do you think that the chemoperception plays a role in all this behavior?
It definitely could. So we did some follow-up experiments. Basically, we put cleaner shrimp in the lab in a little tank and like leaned an iPad against the side of the tank. And you can get them to antennae whip and even try to clean just like rectangles and circles and things going by. And we found that they would antennae whip and try to clean even just those purely visual stimuli. So in the absence of olfactory cues, they would still do all of these behaviors, but not a very high rate. It was something like 25% of cleaners would antennae whip to the purely visual stimuli, and then only about half of those would then go on to try to clean the iPad. So it's possible that, I think it's definitely possible there's an olfactory sensing role for that behavior, or maybe that's even where the signal originated, right? It's originally an olfactory sensing behavior that has since been co-opted to be recognized by the clients as they approach. Yeah.
Okay, thank you very much. Really nice.
Great questions. Thanks.
Miranda has a question.
Yeah, sure. Hi, thanks, Eleanor. That was super interesting. I'm more on the ecology and evolution side, so it's always fun to like hear about all of these vision and perception and all that. I was wondering about the jolting behavior. I thought that was quite interesting because that arrow in your big network was like really high, and so it was like 80% or something. So I was just wondering, like, do you think that that's something that the clients are like, are they getting pinched, do you think, and then somehow they're moving and that's signaling to the shrimp to stop cleaning, or do you think that they're like doing some sort of behavior that then like tells the shrimp like, I'm done, I'm bored, or like I need to move on with my life or something like that? I thought that's interesting behavior.
Yeah, that's a fantastic question. So there are also cleaner fish, and there's a cleaner RAS system that's particularly well studied. And in that system, it's pretty well demonstrated that jolting behavior indicates that the cleaner has basically cheated, right? It's taken a bite of healthy tissue or mucus that's not what the client wants it to eat. And so it's like a painful response to being bitten. But we don't know that this is the case in cleaner shrimp. We don't know what the jolting indicates. And, they're just so small. It's hard for me to imagine that even if they did cheat and take a quote unquote, like as big of a bite as they could out of these fish, that it would mean very much or even cause much pain. But the question you've hit on is exactly one that like came up in my defense actually. And it's puzzled me ever since, because from the data I have, you're right, we can't actually distinguish between the hypotheses that it is maybe cheating, and so shortly after being cheated, the client decides to leave, or alternatively, that it's actually some kind of indication from the client that it's ready to go, because we tend to see it start to happen closer to the end of an interaction, and then after a few jolts, the client will leave. So to really get at this, we have to do something in the lab, probably, artificially parasitizing some clients, but not others, and looking at jolt rates when there's actual parasites to be eaten versus not. And it's definitely an open question in cleaner shrimp for the moment. Yeah.
Super cool. Thank you.
Thanks.
There's a question from Abhinav.
Hi, so do you see how much change is there in the capabilities of this cleaner shrimps after you incorporate motion perception? Do you see a lot of change?
So how much do they vary in like temporal resolution, things like that?
So like if you incorporate how they perceive after, in a video you show a perception of fish after you incorporate how they move in space. How much effect does this have on how they see?
On how the clients view antennae whipping? Yeah, that is something we haven't looked into at all. The sort of either the temporal resolution or motion perception on the part of clients. It could just be that because motion is particularly attention grabbing, it's particularly effective at demonstrating that the client has arrived at an active cleaning station that's currently staffed by cleaners. The other thing, and I don't know, this is tangential to your question, so hopefully you don't mind. But it's been suggested to me that perhaps the fish lateral line is sensitive enough to be actually detecting that motion as a tactile stimulus. And that, yeah, this is something, it's been on the 5-year time horizon for me for more than 5 years now to actually try to look at that. But you can put a shrimp in a tank and do these like particle flow experiments looking at how the water motion actually propagates due to these antennae, right? Like they're really small, but also the fish are very close and the lateral line is extraordinarily sensitive. So it's possible that the actual motion of that antennae is not just a visual signal, but something else additionally. So, yeah.
And also following up from the previous person's question. So do you know how these shrimps detect ectoparasites?
No, we don't. So it used to be thought that a prerequisite to being a cleaning organism was actually having high enough acuity to visually locate ectoparasites. That's probably true in some cleaning systems, like there are cleaner birds, cleaner mongooses, but in cleaner shrimp, I think it's probably tactile. There is the idea that color change behavior might make parasites more visible to cleaners, but I don't have any evidence of that myself. And at least like you can get cleaner shrimp to hop on your hand and like clean off your cuticles and stuff, and they're just tapping all over you. It seems like it's a very tactile process, but that's completely anecdotal. There's no real hard proof there. Yeah.
Thank you.
Thanks.
Thanks. So I'm just gonna, I think Dr. Case can stick around a little bit longer to answer questions. But I just wanted to conclude this session 1, because some people need to leave, and we hope to see you for session 2 in 11 hours from now. Dr. Case, is that right? Can you stick around a little bit longer to answer the rest of the questions? Okay,
Sure.
So with that, thank you all for, it's been really nice to see you. Thank you all for attending, and we hope to see you in the next session. Okay, so the next question is from Sunny.
Yeah, this is a good question.
Hi.
You've hit on the $1 million question. But in case everyone hasn't read it, I'll let you ask it. Yeah.
Oh yeah, okay. So first of all, thank you for your talk. I enjoyed it very well. And it's not the direct topic in your talk, but I've seen the products of acuity view, how you've modeled how different images might view or seem to the visual system of the study subject. And I've noticed that the images are always the same size, even though you model different distances.
Mm-hmm.
And I have been wondering whether it would be possible to somehow take account how the image size will change with the distance.
Yeah, so it's a great question. I get this all the time about acuity view. So basically, acuity view in the mathematics underlying it, and this is very much the Sanka-Johnson realm of this R package, but in the mathematics underlying acuity view, it's accounting for changes in distance when it's spitting out that final product. But you're right, the final product is always the same size. So the way we think about it is, as you move farther and farther away, that same size image that's coming out is just taking up a smaller portion of your field of view. And the reason we didn't write the program to change the image size is because often we were interested in, a couple of orders of magnitude of distance. And so then the program would spit out these images that were like teeny, 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 and some of them that were like absolutely giant, and they were kind of uninterpretable. It's a trade-off though, because I agree that it's a little bit hard to think in your head about, it's uninterpretable when it's a teeny, teeny image, but also now it's just on the user to think of it as being a tiny image. So yeah, it's basically that indeed the size of the output is not changing, but that output is taking up different portions of the field of view. And so I have seen people shrink the images themselves when they come out to sort of account for that or blow them up, but it's something we haven't quite worked out the best solution to. Does that answer your question all right?
That answered it quite well, and I think like you have a very good justification for why not also, and I'm already thinking like this visual attention and stuff like that. So in those cases, those tiny images could like really well display what the animal is actually viewing in the field of view, and how something might be like spot or like viewable, seeable or not. But
Exactly. Like people, I often use acuity view to look at like how does the bug on a tree appear to something that's 10 meters away, and sort of spitting out these like teeny tiny things. And that maybe is really how it looks. So, yeah, there's trade-offs, but.
Thank you.
Mm-hmm.
Okay, we have a question from Alex. Is Alex still here?
Hi, yeah, the question was just prompted by a discussion about the jolting behavior and whether that signals the end of the sort of client-cleaner relationship. And it just made me wonder what happens if the cleaner doesn't get the message and doesn't come back down off the fish. Like, can it get kidnapped, or does it just like jump off and it's fine?
Yeah. Good question. So usually I would say the cleaners hop off first. Like more often the situation is the cleaners have hopped off and the clients just stick around, perhaps out of hope that they'll get cleaned again, but then they don't. I have seen fish try to swim away with the cleaners still on them, but that's kind of rare. one interesting thing that we've seen is that most of these cleaning stations are staffed by multiple cleaners, and there's like one individual who's the largest and does most of the signaling and most of the cleaning. And once that large individual has hopped on and started cleaning, we see some of the smaller individual shrimp hop on and also start to clean. And they're some of the ones that haven't hopped off more often by the time the fish leaves. It's like they're less in tune with the whole system and like how to navigate these interactions. So sometimes the fish will try to swim away with those guys still on board. Actually, I don't know if Laura Baggy is still in here or not, but she has told me about like poking cleaner shrimp off their anemone in Belize, I think, and seeing them get eaten when they're not in the protective sphere of the cleaning station. And so I think it's true. We think there's some special context, some behavioral context to the cleaning station that helps keep cleaners from getting eaten. And they really don't want to leave that area. So I think as soon as the client fish starts to leave, they usually just like hop right off. Yeah, she says I'm here and agree with that. So yeah, I think they're really eager to not go anywhere outside of the cleaning station. But I'm sure they do occasionally get hijacked further than they want to go. Yeah.
Right. Thank you for the answer.
Thanks for the question.
Nico has another question. So now Nico has a chance to talk.
Oh, sorry, different Nico, actually. But
Hi, Nico.
Hi, Eleanor. Just to follow up, I guess, like, it just got me thinking about how do they negotiate when to stop? So any follow-up on that is great.
Yeah, I don't know, but it's a great question, whether it is a negotiation or whether there's one party that's potentially more in control. And I don't have a good answer to that, because I could imagine that either party could be the one who's really driving the end of the cleaning interaction. You know, in cleaner fish, we see that the clients seem to be reasonably in control because they can choose other cleaning stations to go to, and there's really complicated dynamics there, but we just haven't got a handle on that kind of detail yet in cleaner shrimp systems. But I think a lot of it will become more clear when we look more closely at this jolting behavior. And there's some work I want to do looking at, like, is the antennae whipping signal maybe, you know, I think it's intent to clean, but what's driving that? Is it like hunger level? And so you might imagine that if it's been longer since the previous cleaning interaction, the shrimp are more likely to whip their antennae and then maybe to clean for longer because they were hungrier. I'm not sure. It's a really good question, but we just don't know anything yet. There's so many unanswered questions. People should come join my lab.
Great. Actually, the other Nico did have a comment that we could all look at. And then there's another question. Oh, another question from Miranda. She says, is cleaning a scarce resource in the environment? Oh.
Sorry, I wasn't sure if you were going to go to the other Nico's question.
Oh, it didn't look like they had a question. Does Nico Michaels want to make a comment on their observation?
Well, it's not really a question. It's just to show a special example of cleaning by shrimp, where a small group of catfish is being cleaned all at once by a single cleaner shrimp.
Yeah, I was just
the fish are hardly any bigger than the cleaner shrimp. But that's just a fun slide. Nothing else.
Okay, Miranda.
Yeah. So you said at one point that like the clients will sometimes stick around longer and like hope desperately that they'll keep getting cleaned. And then you also had the other diagram where it sounded like even when the clients are posing and changing color, that they actually only get cleaned like half of the time. So I'm sort of wondering, is this cleaning, like, is this like a scarce resource in the environment? Is it limited in some way? Is there like competition over access to the cleaner shrimp or other cleaners, I guess? Yeah, that's basically the question. Is it hard to get cleaned?
Well, I think it depends. It varies a lot. I think there is some competition in that, you know, there are client fish visiting cleaner shrimp all day, nearly constantly. So there's certainly a demand for these cleaning services, but whether that's a bunch of different clients wanting services, or like one wanting them multiple times. I mean, I haven't individually tagged any fish in the wild yet, but from, identifying features on some of them, you can tell that some of them get cleaned, like literally swim in a big circle and come right back and get cleaned again. And so it's possible that certain individuals are seeking cleaning a lot, depending maybe on ectoparasite load or something like that. But whether it's the limiting resource in the environment in general, I only have anecdotal evidence of this, but like at that site in Curacao, it's really kind of a rubble reef. It's not in very good shape. It's like wedged between this thing called Pirate Bay and an old Hilton Pier. And there are tons of cleaner shrimp, like tons and tons and tons. But on some healthier reefs I've been to in Curacao, there's really not as many. So I don't know exactly if that's because the cleaners and their host anemones like this intermediate level of disturbance. Maybe that's a particularly high ectoparasite area for fish. So there's a lot of cleaning to be done. But usually on a given reef, there's multiple species of cleaners operating at the same time. And in a dense area, there's, sometimes cleaning stations are right next to each other or a few meters apart. So I don't know if it's a limiting resource everywhere, but clients do seem to come from kind of far away to seek it out in certain places. So yeah.
Very cool. Thank you.
I think we're good. Did I miss any questions?
Oh, that was fun. Those were really good questions. I know there's a lot of unanswered and very exciting questions. So
thank you so much for staying later and talking to us and for your talk.
No problem. Yeah, thanks for inviting me. This has been a lot of fun and I'm looking forward to session 2.
Thanks a lot, Eleanor. That was great.
Thanks.
Yeah, thank you. Very interesting. I can't wait to learn more about cleaner shrimp.
Yeah, stay tuned. The lab is small and slow at the moment because it's just me, but we're getting there slowly.
Have you already moved there, or were you at Exeter? Is that right?
Yeah, I was at Exeter until the end of June. Got to Santa Barbara in mid-August, so it's still pretty new. But yeah, learning my way around. I have like printer access now and keys to my office, so
important things.
Yeah, right. And the quarter just started, so now there's like students. It's kind of fun being back on a college campus with people. So
yeah, are you teaching this semester?
No, luckily no.
That's nice.
I'm teaching next quarter, so winter I have some teaching to do, but not right away.
Exciting.
So are you all are staying, and Mike, it's gonna be like really late for you session 2, is that right?
No, it's gonna be very early, much worse.
Very early. Okay, so you'll get some sleep and then come back to it.
I would much prefer very late.
Well, it's been great so far. The breakout rooms were so easy to navigate and everything. I really enjoyed being able to hop between the sessions this morning. It's great.
Yeah, it's fantastic. It worked so well. Everyone was using them so well. Yeah, so good.
Yeah, awesome.
Great.
I was afraid to say.
Oh, go ahead, Mike.
Just talking about the conference organizing. We don't have to do that.
Wait, but no, actually, I do want to know how you guys got, like, was this a volunteer job? How did you?
This is Rox's idea.
Okay, that's great.
Rox is the mastermind.
Wow. Cool.
I want to try out Acuity View now too, because I've been wondering about, I study fruit seed dispersal in fruits. And there's like some very large fruits out there that just don't have that much color to them. I've always wondered, like, does the size kind of compensate for the lack of color? But then some large fruits like watermelons have kind of stripes on them. I'm like wondering if that could sort of increase their perceptibility, but then birds have pretty good acuity anyway. So maybe it doesn't matter, but I want to try playing around with it because it sounds fun.
It's fun to play around. And so I would also say that, so Acuity View in R is like mildly inflexible because of the way that R deals with images, but Cedric and then Julian Troshenko built Acuity View and some other techniques for looking at acuity into their Mika toolbox. And that can be a little bit more flexible and like, it doesn't require square images and things like that. So that's another place I would look if you're needing more flexibility than the R package allows.
I'll look into all of it. Yeah. Thank you.
No problem. All right, I should probably go get my day started, but this was a really nice way to start the day today. So thank you all very much.
Yeah, thank you so much for coming. This was
Thanks
super
for coming.
interesting.
Thank you.
All right, talk to you again later.
You guys
Bye,
like
everyone.
Bye.
Oh, everything stopped. Yeah. I think I might leave my computer just running overnight. So that
Are you afraid that everything will fall apart if you shut down
it's
your
not
Zoom
so much
session?
that, it's just that if I shut the Zoom session, then I have to remake all of the
Mm.
breakout rooms at 4 a.m. Which I don't really want to do. So I thought I would just leave it. And it'll be weird because obviously people will join randomly all day and they'll see it all set up. But they've been doing that anyway. So
Yeah.
I just think
Maybe
it's
make
fine.
sure you're not recording all night.
Yeah, who is recording now? I thought I turned it off.
I'm not.
I'm not.
Sebastian's still recording.
No, am I? Oh,
Yes, you are.
Someone is.
oh, I will stop that. Stop.
Yeah.